Okay, everybody, good morning, good evening, good afternoon. This is Dr. Kaz, and I am bringing you another installment to our Fine Anatomy class. And we are going to be discussing Chapter 29, the Development Chapter. So let's get right to it. Okay, so we are going to discuss, all right, the prenatal period. And our prenatal period is going to be the period here, right, in which we just fertilized our oocyte. Right, the sperm has come in, and so this period begins right with fertilization when our secondary oocyte and the sperm have come together for fertilization here. And so we'll see this right around 30 weeks, possibly 40, right? And that's when the prenatal period will end, usually during that time. So in this prenatal period, we have three kind of smaller periods in there. We have the pre-embryonic period, the embryonic period, and the fetal period. So starting off with the pre-embryonic period, this is a 2-week period of time, right, immediately after fertilization. And so we're going to see, right, our fertilized secondary oocyte and united sperm, that structure is going to have several different names. Right, but as soon as fertilization occurs, we start off with the zygote here. And this zygote is going to be kind of like this circular structure, and these cells are going to undergo some rapid division. We'll talk about what that is, the cleavage period there. And we're going to finish up with this structure called a blastocyst. And finally, that blastocyst is going to implant itself onto the uterine wall. So we're going to discuss all that. So then the next period after the pre-embryonic period is, of course, the embryonic period. And so this goes between the weeks of the 3rd week and the 8th week here. And again, we'll see the beginnings of some of our major organs developing, right? A very simplistic form of those organs, because we'll spend a lot of time having those organs go through maturation, development, and we'll talk about that later. But in the embryonic period, now we're referring to our structure as an embryo. And then finally, we have the fetal period. And so that is going to be the time after the 8th week. So that'll be the remaining 30 to 32 weeks. And that's the final time prior to the birth of the baby here. And at that point, we refer to our structure now, our organism, as a fetus here. In this fetal period, we're going to see a lot of dynamic changes occurring in the fetus throughout all the tissues, the tissue development, muscle tissue, all right, connective tissue, including the skeletal system. You'll see certain systems like the digestive system, circulatory system. All of these systems are going to be developing at a phenomenal rate. So we're going to discuss that. So, all right, let's begin here and take a quick peek here at our prenatal period and maturation diagram. And so again, we're going to start off here, all right, with the pre-embryonic period, weeks 1 and 2. And so you'll see right after fertilization, now we're going to have the united egg and sperm start to go through this period of mitotic division. And so we'll discuss this here in a few moments. And then our next stage, which will be gastrulation, excuse me, and then organogenesis, when we really start to see, right, the development of the internal organs, right, and our systems really increase in their complexity. All right, so let's just kind of discuss what fertilization is here and what happens to our newly formed, all right, zygote. So we learned in chapter 28, right, about the journey of the sperm and the journey of the oocyte during ovulation. And then when intercourse occurs, what happens with the sperm and their travel journey towards the secondary oocyte. So when this all occurs, and normally we'll see fertilization occur in the fallopian tube, specifically the ampulla. So if you recall, the ampulla is that portion of the fallopian tube. It's a kind of a widened area of the fallopian tube, right, between the isthmus there and the infundibulum. And so fertilization occurs, all right, when, all right, the two gametes, the secondary oocyte and the sperm, fuse together. And those two gametes are haploid cells, but when they fuse together, the resulting cell is going to be a diploid cell. And so again, at that point, that new formed structure is going to have genetic material from both mother and father. Both parents have now provided that new organism with, all right, genes from both mom and dad. And so at that point, we are no longer haploid. We now have our diploid number of chromosomes. And at the same time, we are going to determine the sex of the organism, whether it's an XX or if it's XY. And then after fertilization, we begin the cleavage period where these cells now, or the new diploid cell, is going to start to rapidly divide. And so this occurs, right, for the majority of the zygote's journey from the uterine tube into the uterine cavity there. And so keep in mind, our secondary oocyte only has, and it's a ticking clock of 24 hours for fertilization to occur. All right. So once ovulation occurs, okay, that the clock is ticking, and that oocyte is only viable for 24 hours. Now, sperm, a little bit longer, 3 to 4 days, but it doesn't matter as much about the sperm because the oocyte's period of time is much shorter. So we have to concern ourselves with that oocyte and what is going to happen, all right, prior to fertilization there. Okay, so let's discuss fertilization here. And there's a couple things to really appreciate what happens with fertilization and really what the sperm have to endure, making it from the vagina up into the uterine cavity and then eventually out to the fallopian or uterine tubes there. They have to go through a rigorous journey there. It's not the best environment for them. One, the acidity of the vagina makes it tough on the environment for the sperm to actually survive. So sperm really don't have a lot going for them during this time. And so there's a process there as the sperm are trying to make their way to the secondary oocyte. They have to undergo this process called capacitation here. And it's kind of like a conditioning there, almost like training for a fight or something, in which the sperm, all right, are challenged to become, all right, ready for fertilizing the secondary oocyte. And so this happens as they're traveling through, all right, the female reproductive tract from the vagina, from the uterus into the uterine tubes there. And so on the outer portion there of the sperm, they have to kind of peel away some layers there, because when the sperm actually have the opportunity to penetrate the secondary oocyte, right, they need to be able to release, all right, the nucleus of the sperm. And so we kind of like almost like an onion, we have to kind of peel the layers away. And so the coating there to the sperm is a glycoprotein coat, all right, with the addition of some proteins there that kind of help provide that covering there. And so we have to remove some of that. So we're kind of exposing the plasma membrane of the sperm. So then when actual fertilization is occurring, where the sperm are trying to penetrate the secondary oocyte's outer layering there of the zona pellucida and of the corona radiata, that they can actually get the nucleus there of the sperm into the secondary oocyte. So this process of capacitation takes a couple hours. And as a result then, right, you'll have to understand that during intercourse and climax, when the male ejaculates, the number of sperm that are released, it's not in the hundreds or thousands, it's in the millions. And the reason why is because, right, only, all right, a couple hundred sperm are going to eventually make their way up to the uterine tubes there. So considering that you know, you're going from the millions to hundreds, you know, that's a significant reduction. So your chances of fertilization decrease several fold. And so they need a little help. Well, as luck would have it, or as nature has planned it, right, the actual secondary oocyte at this period of time is releasing, right, attractive chemicals that kind of guide the sperm, right, towards the target there. So the oocyte is releasing these chemicals, and so the sperm now have a kind of like a homing beacon, and so they are going to start to travel in that direction. And so, as a result, some of those chemicals are going to increase, all right, these calcium channels in the flagellum of the sperm to kind of direct it towards that area there, but also it helps it make it able to undergo that process of capacitation. And we're going to talk about the achromosome reaction there. That's when the sperm actually is trying to burrow its way into the secondary oocyte, and then eventually we yield fertilization from that. And so because of all this, think about it. If you get hundreds of sperm kind of converging on the secondary oocyte, only one of those sperm are going to actually fertilize the egg by design, okay? Because if you get more than one, if you get polyspermy, then you can run into some genetic issues there in which, all right, the zygote becomes unviable and eventually it just won't live, right? So that's why one sperm, all right, per egg. So here's kind of what happens with fertilization. And I'll show you in picture form here in a moment, but let me just walk you through some of these steps here. All right, so of course, if you recall the structure of the secondary oocyte, you've got on the outside of the oocyte immediately, you've got the zona pellucida, that's that glycoprotein structure. And then outside of that, we've got the corona radiata. And the corona radiata is going to have all of those granulosa cells that are just sitting there, right? And at this point, when the sperm come in contact, because you can have multiple sperm making contact with the corona radiata, but only one's going to get through. And so basically the sperm are going to try to push their way through those cells there of the corona radiata and try to enter into the zona pellucida. Right, so basically that first phase there is corona radiata penetration, and that's when the sperm are pushing their way through those cells. Well, then you get one of the sperm that makes it to the zona pellucida, and as soon as that happens, the head of that sperm embeds itself into the zona pellucida, and it undergoes the achromosome reaction. That's the tip of the sperm there. And the achromosome releases these digestive enzymes that pretty much break down the zona pellucida in that immediate area. And when that occurs then, right, as soon as that happens, which is kind of like a cool protective mechanism that the secondary oocyte has, right? As soon as that happens, right, the secondary oocyte, right, causes that zona pellucida all around the oocyte to become hardened, thus making it impossible for other sperm to penetrate the zona pellucida. So this is the reason why only one sperm can fertilize the oocyte. So like I said, if you have several sperm that do that, we call that polyspermy, and this causes some genetic or chromosome abnormalities. And normally that causes the zygote to become, all right, inviable, and it end up dying there. Interesting process here. Okay, so on to the next phase here of fertilization. And this is actually where we see, right, the sperm and the secondary oocyte, both of their plasma membranes now fuse. And as a result of this, we are going to see now, right, the nucleus of the sperm is going to enter into the oocyte. And as a result of that, right, if you recall from chapter 28, the secondary oocyte is still stuck in its second meiotic phase there of division. And so because of that sperm nucleus entering into the oocyte, that causes the secondary oocyte to complete its second meiotic division. And so at that point, we now refer to it as an ovum. And from this point, the nucleus of the sperm and the ovum, they come together and fuse. And so keep in mind that each of those structures, the nucleus of the sperm and the ovum are only haploid number in chromosomes. But once they come together and fuse, they are no longer, all right, a haploid number of chromosomes. Now they've restored the chromosome count to the diploid chromosome count. And as a result of that fusion there, right, we now have ourselves a zygote. So once we restore, all right, the diploid number of chromosomes, all right, that structure is now referred to as a zygote, and that is one cell with diploid number of chromosomes. All right, so here in picture form, all right, as promised, just to kind of walk you through so you can kind of see what I was talking about, our phase one here, the sperm have approached our secondary oocyte. And of course, secondary oocyte kind of has these two layers. The outer layer, that's the corona radiata, and then that inner layer, that's zona pellucida, that's that glycoprotein layer. So the sperm first have to work their way through these cells here on the zona pellucida. Right? And so as soon as they penetrate, right, the corona radiata, that's the completion of phase one. And so that sperm that does that and it beds itself into the zona pellucida, now we undergo phase two, that a chromosome reaction where these digestive enzymes leak out of the head of the sperm here, and that helps it to make its way through, right, the zona pellucida here to penetrate that. And as soon as that will happen, all right, as the sperm makes itself to the zona pellucida, we get that hardening. So you can see in this picture here, there's other sperm over here, right? So these other sperm are prevented from entering into the oocyte because of that chromosome reaction, which causes that hardening here of the zona pellucida. And that's phase two. Then finally, phase three, right, once the sperm makes it to the plasma membrane here of the secondary oocyte, then we get a fusion of both of those structures, and then the nucleus here of the sperm, right, moves into the secondary oocyte here, and that ovum now, or excuse me, the secondary oocyte completes its second meiotic division, and now we have what's known as the ovum pronucleus and the sperm pronucleus, and both of those will fuse together. And once that occurs, now we have a zygote. So think about it. Haploid chromosomes in the sperm pronucleus, haploid chromosomes in the ovum pronucleus. When they both fuse together, now we restore that diploid number of cells and we're good to go. Okay. So now, all right, we've completed fertilization and we have a zygote. So the next part, now we're done with meiosis. So we don't have to worry about that, not that we were worried to begin with, but now we're going to see a series of mitotic divisions, right? So where we have one cell, we're going to start to have 2 cells, then 4 cells, then 8 cells, then 16 cells. And so important concept to understand during this time here, this cleavage period of time, right? We obviously are going to get an increase in the cell number, but not in the cell size. Okay, so number increases, but not the size of the actual structure itself, of that zygote. And so really the only time we're going to see an increase in the size is going to be just when, all right, the zygote, but it's not called a zygote at that time, it's called the blastocyst, when it implants itself into the uterine wall, it burrows itself in, then we're going to start to see, right, a significant increase in the size of that structure, but not until, all right, implantation occurs here. All right, so we see this cleavage period occur, right? So you go from 1 cell to 2 cells to 4 cells to 8 cells. So before we get to that 8 cell, all right, stage, the cells that are being produced, they're not tight together like you would see in epithelium, for example. They're loosely, all right, bound to one another. It's not until, all right, we get to the 3rd cleavage division, Okay, so that would be, we go from 1 to 2, that's the first one, from 2 to 4, that's the second one, and from 4 to 8. All right, so after we get the 8 cells there, we have them loosely touching one another. Right, after that, then we get the phenomenon of called compaction, and that's when the cells now become tightly packed. Right, so they will have this tight contact between themselves, and we call that compaction. So after we go from that 8-cell structure, and then we develop into a 16-cell through, all right, the mitotic division, we give that structure a name, and we call that the morula. And now we're going to see, instead of just a glob of cells, we're going to see some new developments occur. We're going to start to see a cavity fill or develop, and there's going to be some fluid in there. And so we refer to that cavity as the blastocyst cavity. And when that occurs then, right, we no longer call it a morula. We are now going to call it the blastocyst. I'll show you pictures of this in a moment. So the blastocyst, right, has 2 structures to it. The first structure is called the trophoblast, and that's an outer ring of cells that are going to be surrounding this cavity. And then you'll notice off to one side, you'll see a clump of cells off to one side, right? And those clumps of cells, that's called the embryoblast. And the embryoblast is going to actually be the embryo, okay? Eventually, which will become the fetus and the baby. Right, the trophoblast, and we'll talk about what the chorion is later on, but that's going to be the beginnings of our placenta, right? That comes from the trophoblast. The embryoblast is going to be the embryo proper, and the cool part about it is all of those cells that are found in the embryoblast and this little clump off to the side here, those are pluripotent cells, which means they can develop into any type of tissue in your body. They can develop into epithelial cells. They can develop into connective tissue. They can develop into muscle or nervous tissue. So those are what we refer to as pluripotent cells. So here we can kind of see, right, during that process, that cleavage process here, you will see, right, immediately after fertilization and union of the egg and sperm nuclei here, those pronuclei, we now have our zygote. So we have this one cell. And then we start to enter into that mitotic division period. Right, so that one cell divides into 2, then 4, then 8, and then we get 16 cells. Then we change the name and we call that the morula. And then the morula will start to develop a little cavity on the inside, and we call that the blastocyst. So you have this cavity here filled with fluid, that's the blastocyst cavity. Then you've got this ring of cells on the outside, and that's called the trophoblast. And then you have our embryoblast, and the embryoblast, this is what is going to develop into the fetus eventually, AKA the future baby. And as you can see, during that time, that zona pellucida is still on. Look, as the zygote is moving through and undergoing cleavage, we still have this hardened, all right, zona pellucida. So no more sperm can get in there because there's probably still some sperm hanging out, right, in the uterine tubes, in the uterine cavity. And so this kind of gives it a shield to reject any sperm trying to get in there. And so once we enter into the blastocyst development, then that zona pellucida starts to degenerate and kind of fade away. Right, so this slide here shows us kind of where along the fallopian tubes, where these processes are occurring. So as you recall, all right, you get ovulation when our follicle has released the secondary oocyte into the infundibulum here of the uterine tube. And then now we see that secondary oocyte start to travel through the uterine tube from the infundibulum into the ampulla here. And then typically this is where fertilization is going to take place. And then from there, right, you've got about a 3 to 4 day journey from here all the way to the uterine cavity. And that's where we're going to see that cleavage process occur. So we start to get to the 2, 4, 8 cell stage, and then eventually we get the morula stage, and then we start to move into our early blastocyst stage here into the uterine cavity. And then our blastocyst will find itself, right, implanting onto the lining of the endometrium here. And I mean, it literally burrows its way into, like a crab does in the beach, all right, under the sand. That's what happens here, right? This blastocyst will burrow its way into the wall here of the uterus. And we'll talk about all that, what happens during implantation. Okay, so now let's get into the implantation of our future fetus, aka baby. So the blastocyst now has left the fallopian tube and has entered into the uterine cavity. And this takes about a week. I mentioned before, usually that journey of the oocyte down the uterine tube takes about 6 days. So a lot happens in those fallopian tubes. By the time the blastocyst has made itself found itself in the uterine cavity, it no longer has that zona pellucida. That thing's pretty much gone. And now it's a structure in search of a home. And so it'll come in contact with the endometrium, and that is the innermost lining of the uterus. And we discussed there's 2 layers to that. And the outermost layer is what we refer to as the functional layer. That's what the estrogen, the progesterone, we're spending all that time preparing the uterus for that layer to become thicker, to have more blood vessels and uterine glands there. So around day 7, our blastocyst finds its way implanting onto the endometrium. And so what we're going to see here is we're going to see the trophoblast. Remember, that's that outer circular ball-like structure. It's going to divide into 2 layers, pretty much where the blastocyst has embedded itself or made contact on the uterine wall. And so those 2 layers, we have the cytotrophoblast, and that's the inner layer, meaning that's the layer that's closest to the blastocyst. And then you have the syncytiotrophoblast, which is the outer layer, and that's kind of like this area right here. And so by day 9, like a crab on the beach, right, our blastocyst has now burrowed itself completely into the uterine wall. And why is it doing that? It's looking, all right, for those nutrients, right, that it needs to sustain, right, its life. And so where is that located? In the functional layer, but deep in the functional layer, we'll have, right, those uterine glands there, which are going to provide all of those nutrients. So this is a good time to talk about HCG, human chorionic gonadotropin. And HCG is commonly the hormone that is used in, well, not used in, but is detected by most pregnancy tests. And so the syncytiotrophoblast, that's that outer layer there, which is displayed right here, okay, that syncytiotrophoblast. You can see it progressing. So this layer here is going to produce that HCG. And so this lets the female reproductive system know, hey, you know, mission accomplished. We've landed on the moon. But in this case, implantation has occurred. So now that allows a communication over to the ovary where the corpus luteum is. The corpus luteum is that remaining follicular structure in the ovary that released that secondary oocyte. And it tells the corpus luteum, hey, don't degenerate. We need you, buddy. And we need you to keep producing estrogen and progesterone so we can maintain and build that uterine lining here, okay, so we can sustain our embedded blastocyst. So we can, in pregnancy tests, we can see this hormone in the urine there by the second week. And so this process goes on for about 3 months. So the syncytial trophoblast is going to be producing the human chorionic gonadotropin hormone, which is going to tell the corpus luteum, please don't break down, don't degenerate. We need you to keep producing estrogen and progesterone, all right, to build that uterine lining here. Eventually, though, right, we won't need that to keep occurring, because then we'll have a functional working placenta that will take over the role of the corpus luteum. But until then, for 3 months, that corpus luteum is going to be producing the HCG. So we can build and maintain that functional layer, that uterine lining there, to help sustain, all right, our blastocyst, soon to be known as something else here shortly, right? But we want to keep it alive. And that's what we pretty much do. So here in this picture, you can see, all right, how we're getting the kind of progression here. Here comes the blastocyst with its 2 layers, the trophoblast, the ring-like structure, and the embryoblast. And then here we have the functional layer of the endometrium, and you can start to see these uterine glands here. We're going to start to see the formation of some primitive blood vessels, right? But that blastocyst comes rolling along. It embeds itself onto, or implants itself onto the endometrium. And then we can see the cytotrophoblast and the syncytiotrophoblast form. And as that's occurring, the structure starts to move its way internally, burrowing its way further and further and further into the functional layer of the endometrium until it's completely covered here. Now you'll notice during that implantation period, and we'll discuss this here in a moment, we're starting to see some more differentiation. We're starting to see some changes in our structure here, right? For example, right, you start to get, right, the bilaminar germinal disc here, which is just the creation of 2 separate cell layers here. We'll talk about that in a second. We're seeing some more cavity formation occurring. So a lot is going on here, and a lot happens. Look, we're only at day 9 by the time we get complete, all right, implantation and embedding of our structure here. And it's quite an amazing process. All right, so this slide here is showing us the hormone levels during pregnancy. So close to the end of the 2nd week here, during implantation, you'll see how HCG just spikes. And it stays like this, you know, in high levels for 3 months, because again, it is going to tell the corpus luteum, hey, I need you to produce, all right, both progesterone and estrogen to build up that uterine lining. And then by the time the placenta is ready to go, and that's usually, you know, at around the 3-month mark, our HCG levels kind of decline, but that's no problem, because now we have the placenta producing enough progesterone and estrogen to take over for the corpus luteum, and then the corpus luteum can go on and degenerate, and then we don't need it anymore. And so then it'll transition into the corpus albicans, then eventually, all right, into scar tissue, and it'll go away. All right, so around day 8, we are now, all this time, we've been talking about the trophoblast, how the trophoblast, in case you forgot, the trophoblast is this outer layer here. And so we see how it differentiates into these 2 layers, the cytotrophoblast and the syncytiotrophoblast, right? But we really haven't talked about the embryoblast. So we're going to do that now. All right, so the embryoblast now is going to give us, all right, some differentiation and form those 2 cell layers that we're going to call the bilaminar germinal disc. So the layer that's closest to the blastocyst cavity, and that's that original cavity that we see right here. This is the blastocyst cavity, right? We're going to call that the hypoblast. And then the layer that is going to be adjacent to the amniotic cavity, that's that newly formed cavity here, right? You can see this nice little cavity forming, right? that is going to be referred to as the epiblast. And so together, we refer to those two layers as the bilaminar germinal disc. All right, so now as the formation of the bilaminar germinal disc is occurring, we're going to see the development of what we call the extraembryonic membranes. And there's three of them, right? So there's three of them. And so these extraembryonic membranes, they're going to be these membranes that pretty much are going to, one, protect the embryo, but maintain and nourish the embryo. You can see their role here. And again, one of these membranes will eventually become the placenta, and the placenta kind of governs all of this stuff, right? So we're going to see, right, nutrition, of course. We need nutrition for this developing embryo here, right? So it doesn't degenerate and die. Of course, we're going to need gas exchange to occur, right, for aerobic cellular respiration. And of course, right, all of that is going to cause metabolism processes to occur. And so, of course, when we have metabolism processes occurring, we're eventually going to have waste product development. So, of course, we've got to get rid of the waste. So these extraembryonic membranes are going to help with this. So the first of the extraembryonic membranes is going to be, right, the yolk sac. And so the yolk sac, this is the first one that develops, right? This one we're going to see, all right, occur with the hypoblast layer. And now we're familiar, maybe some of us aren't, but a yolk sac, when you've seen an egg or in some of the other egg-laying species on the planet, the yolk sac is usually going to be, all right, the sac that surrounds the yolk, and the yolk is usually where the developing organism is going to get, all right, a lot of its nutrients. Well, in this case, that does not happen with humans, right? It does not store any yolk, right? So what the yolk sac is going to be utilized for is for early blood cell and blood vessel formation. And we'll see more of that later on, all right? So again, don't think of it as the traditional yolk sac that you would see in other species in which the developing embryo is going to be getting its nutrients from there. That doesn't happen in humans, all right? But what this yolk sac will do is eventually we're going to see, all right, development of early blood cells and our blood vessel formation, right, for, all right, our circulatory system there. The second extraembryonic membrane is the amnion. So some of us may have heard of amniotic fluid, right? So this amnion, and I'll show you a picture here. This, all right, is going to be continuous with that deeper layer, the epiblast layer. And so eventually what we'll see is the amnion will eventually surround the entire embryo. And again, we're going to be dealing with that fluid-filled, all right, cavity there, the amniotic cavity. And so in several weeks later, if someone decides to do what's known as an amniocentesis, if they want to check on any possible genetic abnormalities that the developing embryo may be having, okay, they're going to go and stick a needle into the amniotic cavity where there's amniotic fluid, and they'll sample that and just basically kind of check out one, right? They can see the sex, right? And they can also check if there's going to be any chromosomal or genetic issues happening there, right? But the purpose of the amnion, right, when it's filled with that fluid, is it essentially prevents the embryo from desiccating or from drying out. So the amniotic fluid is just going to be bathing the embryo and prevent it from drying out and allowing it a good environment, all right, for it to continue on with its development. All right, our third extraembryonic membrane is the chorion. Right, and so this one is going to eventually be the outermost embryonic membrane. That's important because eventually the chorion is going to form our placenta. So it's important that you know the chorion comes from the syncytiotropoblast cells and the cytotrophoblast cells. So those cells will eventually form the chorion, which will then in turn eventually form the placenta. And so they're actually intermingling with that endometrium, that functional layer there in the endometrium. And so once the placenta forms, like I said a little bit earlier there, that's where we're going to see nutrient exchange between the embryo and the mother. So mother's side of the placenta will provide like oxygenated blood, for example, whereas the embryo side of the placenta will provide like carbon dioxide. And so we'll see an exchange of respiratory gases across the chorion there, eventually the placenta there. So speaking of the placenta, let's talk a little bit about the placenta here. Very important structure. Okay, so the placenta, an amazing structure. First, highly vascular structure. Makes sense because you have a developing organism that is going to be undergoing, all right, a lot of tissue differentiation, and so it needs a lot of nutrients from the blood. So it only makes sense. So the placenta is going to facilitate, right, exchange of these nutrients, of these respiratory gases, of any wastes that are being produced from these cells as they're doing their metabolic functions. And so it's essentially an exchange site, right, between mom and fetus. And in addition to that, it's also going to play, all right, a site for the exchange of antibodies from mother to fetus. So it helps with immune function. And also, of course, we cannot forget, it essentially becomes a gland in which it produces estrogen and progesterone. It's taken over that role from the corpus luteum. And so now it is going to do it itself so it can maintain and build up that uterine lining there in the functional layer of the endometrium. And here's the cool part. All this happens second week. We're starting to see this happen in the second week of development. Amazing. It's amazing stuff here. So when we're looking at the placenta, we have two parts. We have our fetal portion, all right, and we have our maternal portion. And the fetal portion comes from the chorion. Mentioned that before, which is the syncytiotrophoblasts and the cytotrophoblasts. And the maternal portion comes from that endometrial layer, the functional layer there. So during the formation of the placenta, we are going to develop this structure called the connecting stalk. And like the name says, all right, connecting, it means it's going to connect, all right, some structures to one another. And so it is going to connect, all right, the embryo to the placenta. Eventually, this will become the umbilical cord. And the umbilical cord contains the umbilical arteries and the umbilical vein. All right, so this is the early part of that umbilical cord that we're familiar with. That is that little, well, I shouldn't say that little cord, but that cord that comes off of your umbilicus, your belly button, and attaches onto the placenta. So that's the early part. So another important placental component is the chorionic villi. These things are cool. So these chorionic villi are these, what we call these finger-like structures. We're familiar with villi. We saw some villi, like the intestinal villi and the digestive tract. Similar to that, all right, so these finger-like structures are going to contain all these branches of the umbilical vessels here. And so they're going to have, all right, the umbilical blood vessels, and I know I'm drawing this, and there's actually a slide here that has some better drawings, and you'll see all these blood vessels in these structures here. And so they come off of the umbilical vessels that are going to be supplying, all right, blood to the embryo. It's important because this is where we're going to see exchange of gases and nutrients, all right, between the mother and the embryo. So of course, here in the functional layer of the endometrium, that is where the mother's blood vessels will be present. And here's the cool part. I love this. I love this, love this, love this, right? there is absolutely no mixing of fetal blood and maternal blood. It does not mix. All right, so how do we get an exchange of gases and nutrients and wastes? Well, the bloodstreams are so very close to one another that they can actually diffuse across the tissue layers. And I'll show you a picture here in a second. So we'll get oxygen diffusing from the maternal blood to the fetal blood. And that's based on gradients. Maternal blood will have a higher oxygen content gradient than the fetal blood. So obviously oxygen will want to go from the maternal blood to the fetal blood. And vice versa when it comes to carbon dioxide. Same thing. You'll have a higher level of carbon dioxide in the fetal blood and a lower level in the maternal blood. So carbon dioxide will want to diffuse from the fetal blood into the maternal blood. I'll show you a picture of that in just a moment here. All right, so some of the things that we notice with the placenta during its development, all right, is that one, it does a lot of its development during the fetal period, as it should, okay, because we got to hurry up and get this thing ready to help sustain our fetus, all right, for the eventual birth of that organism here. So we're going to see it attached very firmly to the wall of the uterus. And then at the time of delivery, once the baby is born, then the job's not over yet. We still have to remove the placenta from the uterus. And so the uterus will contract and expel, all right, the placenta from the uterus, and we refer to that as the afterbirth. So keep in mind, like I said before, this structure is a selectively permeable structure specifically for respiratory gas exchange. Right? Simple diffusion allows the movement, all right, of gases to freely pass, right, between maternal blood and fetal blood. But it's going to block other things, harmful things, bad things, microorganisms, very bad, bacteria, all right, parasites. Those are things that are going to be blocked, not all, but most will be blocked. And then also we are going to see, all right, certain maternal hormones from crossing, right? We don't want to interfere with the development of our organism, so we only want to allow certain things to move across that membrane barrier there, that placenta, right? Because some are beneficial, we want those to pass, but some are harmful, and we don't want those to pass. So you can see the list of things here on the screen, right, of substances that are harmful, right? Obviously, viruses, not good. So we want to prevent, all right, the ability of viruses, bacteria, certain pathogens, drugs, alcohol, and toxins from being able to cross. Unfortunately, we can't do that for everything, but it does it for many things. If we're unable to, all right, prevent the movement of those substances across the placenta there, then we can actually start to cause some birth defects, or worse yet, death, which is not good. That's why a lot of medical professionals will really strongly urge women, if they are a smoker, stop smoking, right? Nicotine can cause vasoconstriction, and vasoconstriction decreases blood flow. Not good. Same thing with certain drugs that can cause certain genetic issues. Also the same thing with alcohol. Fetal alcohol syndrome, all right, is a condition which you'll see in some children when they're born, and you can get some physical deformities from it, but more notably, you'll see some mental deficiencies in those children that have fetal alcohol syndrome. All right, so this picture here is showing us now we're at week 3. We have the development of our extraembryonic membranes here. And so you can kind of see, here is the amnion, and then you can see the amniotic cavity right here where we'll have the amniotic fluid. And then we have our bilaminal germinal disc here. That's the future embryo, all right, the actual fetus and then baby. And then below that here, you can see the yolk sac. Then we have the beginnings of the connecting stalk, and that'll eventually one day form the umbilical cord. But you'll notice all around, all right, this structure in the center, we have the future placenta here, made up of the chorion. Remember the chorion, the cytotrophoblast, and the syncytiotrophoblast. And then you can see the functional layer of the endometrium here. So zoom in a little bit here. You can really start to see here with the chorion, we're starting to get those villi structures here, all right, forming, and that's where we're going to see the site for exchange here of nutrients and gases. So let's zoom in even closer. All right, so here you can see, all right, how we have our chorionic villus here. At the bottom of the screen here we have the umbilical arteries and vein. And then you'll see at the top of our screen, we have the maternal artery and vein. Now notice, if you look at the picture here, there are no actual physical connections between any of the maternal blood vessels and the embryological or umbilical blood vessels, right? They're close though. They're very, very close. And so what we're going to see is, all right, because they're close proximity, right, we'll have not a mixing of the blood, but we'll see we can get a diffusion of nutrients and of respiratory gases, all right, across the chorionic villus here. And that, again, we'll see that in the placenta. So this picture here kind of shows us, we see quite a bit of the chorionic villus here, and we're going to see our structure start to move back out into the uterine cavity here. But we'll talk more about that later on. This, of course, happens late into week 4. So a lot has happened in the first month of development. All right, we're starting to establish, all right, a blood delivery system here, okay, to our developing embryo. All right, we're starting to see the formation of extraembryonic structures here, and we're starting to see now differentiation in tissues. A lot of this very important stuff. So I'm going to stop here, all right, for this video here. I hope you enjoyed it, and just wait for the next installment. Have a great day.